you very much.
Thank you. Can you see it?
Yes, we can see it, Rocío. That's perfect.
Okay. So first of all, I would like to thank Zubair and Richard for inviting me to talk today about the slow release devices. And we talk about diseases of the posterior segment of the eye. All of them are devastated. And among them, diabetic retinopathy, AMD, and glaucoma are among the leading causes of blindness. These pathologies have in common that they are chronic and multifactorial. The site of action is the retina or close to the retinal tissue. All of them are related with visual impairment and blindness, and it's necessary to reach therapeutic concentration of the active substance in the action site during long term. This is a problem, a real problem, because there are effective barriers in the ophthalmic administration. So if you use just the topical administration, the cornea, and if you would like to use systemic administration, the blood aqueous and blood retinal barriers did not allow to reach effective concentration of the therapeutic agents. So for this reason, frequent intravitreal injections are needed with secondary effects and potential toxicity. This is the reason why intraocular drug delivery systems are developed. Attending to the composition, they are composed by one or more biomaterials combined with one or more active substances. And attending to their structure, they are reservoir if the active substance is surrounded by a polymer or a mixture of polymer, or the drug can be dispersed in the polymeric network. According to their size, they can be nanosystem from 1 to 1,000 nanometers, microsystem from 1 to 1,000 micrometers, and implants higher than 1 millimeter. Just for long-term delivery, implants and microparticles are the most adequate, as they are able to release the active substance during long term due to the low surface area compared with the nanoparticles. Ra gave us yesterday an idea of the different implants that are in the clinical practice. There are several non-biodegradable and several biodegradable devices with the different times of releasing the drug. I'm going to talk today about microparticles. Attending to the composition, they can be biodegradable or non-biodegradable. Attending to their structure, they can be matrix structure or reservoir structure, receiving the name of microspheres or microcapsules. We are very interested in this kind of drug delivery system because they have a lot of advantages. The first one is that they can be administered as conventional injections, just using small gauge needles from 30 to 32 gauge. They are able to release the active substance during long term, as I said before. Depending on the size, they can give different release profiles that is very useful. There is the feasibility to include more than one active substance in the formulation that is very interesting for multifactorial diseases. The ophthalmologists can personalize the therapy because depending on the amount of microparticles, different doses of the active substances can be injected. And depending on the biomaterial, the particles can suffer aggregation in the site of administration, behaving like an implant. Remember that Rach told yesterday that one of the problems of the particles are that they can float on the vitreous. So if they aggregate, is better because the vision is not interfering. Furthermore, the particles can be injected through different routes by subtenon, subconjunctival, intravitreal, subretinal, etc. Among the biodegradable polymers, the polylactic co-glycolic acid, the PLGA, is the most employed for preparing the particles, the biodegradable particles, because as Rach already told yesterday, there are implants approved just for clinical use by the regulatory agencies. This polymer is compatible, and the degradation rate can be modulated depending on the PLA-PGA ratio. The PLGA 50-50 is the most biodegradable. And for the eye, the low and medium molecular weight is usually employed. In the development of biodegradable microspheres, there are a lot of factors that we have to take into account. First of all, the pathology. Second, the physicochemical, biopharmaceutical, pharmacokinetic properties of the active substance. Of course, it's different that if you have to encapsulate a biotechnological product at a low molecular weight substance. For sure, the tolerance of the system. Always biocompatible polymers have to be in use, and a high encapsulation of the active substance must be performed in order to inject the lesser amount of polymer. The particle size is important because depending on the size, different needle size are used. In our experience, the particles from 20 to 40 microns are the best because they can be injected using 30 to 32 gauge needles, and they provide good release rate of the drug. Of course, sterilization, depending on depending on the biomaterial and active substances, and usually gamma irradiation is used. We have developed a method in which the device, the particles are sterilized under low temperature conditions, and then the release of the drug is not changed. There are different strategies according to the therapeutic objective, and I'm going to talk today about the encapsulation of the neurotrophic factor GDNF with vitamin E, that we were able to provide long-term delivery, just 19 weeks delivery of the GDNF, and a combination of dexamethasone, melatonin, and CoQ10, just for the treatment of glaucoma. I'm going to talk today about glaucoma. Glaucoma is a multifactorial disease. The most common is the primary open angle glaucoma. It's progressive, and there is an atrophy of the optic nerve and degeneration of retinal ganglion cells and their axons, as Zubair just explained yesterday very well. So in some patients, an IOP increase is performed due to the anomalous renovation of the aqueous humor. But it's very interesting that there are patients with normal values of intraocular pressure that develop glaucoma. For this reason, the increase of intraocular pressure is considered the main risk factor, and the glaucoma treatment is directed to the control of IOP in the patients who have increased values of this parameter, and neuroprotection that is interested for patients with high and low values or normal values of intraocular pressure. In order to develop a formulation just to be for neuroprotecting just the retinal ganglion cells and other retinal structure, we had to take into account that neurodegeneration, as already Zubair explained yesterday, is sequentially. This means that there is a primary axonal damage, then death of the damaged neurons, and finally there is a damage and death of neighboring neurons in the process known as secondary degeneration. Also, we have to take into account that the degeneration is multifactorial. It's a multifactorial etiology in which excitotoxicity, oxidative stress, inflammation, mitochondrial dysfunction, or protein aggregation take place, among others. So our hypothesis is that a combination of neuroprotective agents in low doses maintained during long term results effective if the neuroprotective therapy in glaucoma. So for this reason, we choose the biodegradable microspheres, including a combination of active agents, and we work for a controlled release of the active substances during long term. There are different therapeutic targets in the neuroprotection in glaucoma. Just can be prevention of the toxicity of the ganglion cells induced by glutamate, neurotrophic factors, anti-inflammatory agents, antioxidants, anti-apoptotic, or inhibition of protein aggregation. So we work with different strategies, just encapsulating different agents in combination or alone. And I'm going to talk today about the combination of the glial derived neurotrophic factor, the GDNF, with vitamin. In this case, we encapsulate this biotechnological product, and we develop a novel method just to preserve the activity of the biotechnological product. How can we do that? Just by suspending the GDNF in vitamin E. And thanks to this technology, just the GDNF is included in the formulation as solid, and the first time that is dissolved is from the particles. So this avoids more of the problems associated with the stability of the proteins inside the particles that Zubair, Ra told yesterday. So the particles were able just to release the GDNF, and as it can be seen, the particles change the shape, just disappearing after delivering the drug. So in collaboration with Michael Young from the Skepenside Research Institute for Harvard Medical School, we tried this formulation in an animal model of glaucoma in rats, just creating an increase of the intraocular pressure with several injections of a hypertonic saline solution. And as it can be seen, the model is able just to promote an excavation of the optic nerve that is seen here. And we injected the particles by intravitreal injections 1 week after the first episcleral injection. And we work with 5 groups of animals, just the non-treated that were used as control, just the PLGA microspheres loaded with a combination of GDNF and vitamin E, the GDNF alone, the vitamin E, and the non-loaded PLGA microspheres. We evaluated the effect 11 weeks after intravitreal injection of the microspheres, just by comparing the axon number of experimental eye with the axon number of control eye. We got a really nice results with this strategy, as 72% of survival were obtained in comparison with the GDNF alone, vitamin E, or the Blanch particles. So the next step was to demonstrate that the GDNF was maintained in the vitros for a long term, and in collaboration with Dr. Elena Garcia and Dr. Luis Pablo from Zaragoza, we injected the particles, 2 milligrams in this case, in the vitreous of rabbits. And then we could observe that the GDNF was, from the particles, was higher, the levels were higher than the one observed for the unloaded microspheres and compared with the basal levels, just demonstrating that the GDNF could be maintained for a long term. 6 months is a long term for this kind of devices. One of the last studies that what we have done was the combination of dexamethasone, melatonin, and CoQ10. In this case, we like to demonstrate that the combined therapy is useful in low amounts and maintained during long term. And in the efficacy studies, we compare the multiloaded microspheres with a mixture of microspheres loaded with a single active substance. The idea of this work was to demonstrate that it's better to use a multiloaded particles than a combination, because the biomaterial that you have to use when you would like just to inject a mixture of microspheres is sensibly higher. So for this reason, we prepared the multiloaded particles, loaded with dexamethasone, CoQ10, and melatonin, that the drugs were released simultaneously. And always we developed just the single loaded particles, loaded only with dexamethasone, only with melatonin, and only with CoQ10. So for our strategy, only 0.1 milligram of microspheres were injected, and then we calculate the amount of a single amount of the particles loaded with a single agent needed to provide the same dose of each drug. And the whole amount resulted 0.233 milligrams of microspheres, more than two-folds higher. So we administer the same doses of active substances and different amount of the microspheres. And in collaboration with Dr. Francesca Cordeiro from the University College of London, we try just this formulation, this different formulation in rats with episcleral injection of a hypertonic saline solution. But in this case, only one episcleral injection was used, and we promoted just an increase of IOP, and then the normal values of this parameter was achieved at 21 days after the episcleral injection. In this case, the particles were injected just at the same time of the episcleral injection. We work with 5 groups of animals, just the multiloaded microspheres, the combination of the single loaded particles, non-loaded microspheres, and we use 2 controls in this case, just the animals without pharmacological treatment and producing an increase of IOP, and healthy animals. And the evaluation was performed 21 days after microspheres administration. The retinal ganglion cells density was calculated, and as it can be seen, yes, the animals that with the injury without treatment, the density is lower than for the healthy animals, and the mixture of the particles for the single loaded particles were not able just to promote just a recovery of the retinal ganglion cells, just producing the same effect that the empty particles. And our formulation was able just to promote protection of the retinal ganglion cells, as the values of the retinal ganglion density resulted the same that for the healthy animals. So we can conclude that the microspheres represent an alternative to repeated intraocular injections, that no surgical procedures are needed. They disappear from the site of administration after delivering the drug. They are able to promote drug release for several weeks, even months. They are able just to release low amounts of the therapeutic molecules useful for chronic diseases. And encapsulation of more than one drug is feasible, just being useful for multifactorial diseases. They are useful for personalized therapy and useful for retinal repair. Zubair, if it's possible, because the talk I prepared that ends here, but according to the talks yesterday, I would like to extend for 3 minutes the talk, if possible, because
yes, you may. Yes, you
may. Okay. So yesterday we have a very interesting talks, and I think there are questions to be solved, very interesting, that we can be solved in the Orbital project. And one of them is the acute versus chronic damage. It's totally different. And in this case, we have to use different therapeutic approaches. So we have to study that. And I'm going to talk just for a few minutes about one study that we did just several years ago, just trying to evaluate the efficacy of microscopes loaded with ketorolac, an anti-inflammatory drug. And the animal model we use with the optic nerve axon. And in this case, we prepared the ketorolac-loaded PLGA microspheres that were released for 60 days. And we'll work with different strategies, with different treatments. Just we injected ketorolac solution. We injected ketorolac PLGA microspheres, and we combine the treatments just by injecting the ketorolac solution plus the ketorolac PLGA microspheres in this model. The evaluation was just, or the injection was for all the treatments at the same day of axotomy and 7 days before axotomy, in order to see if there was any protection of the before the injury. And the quantification of the retinal ganglion cells was performed always 7 days after the optical the axotomy. So we talk about short term, when the injury was performed at the same time of the axotomy, and long term, just 7 days before. So look at the results. Just the arrow seems long term, and the sign short term. We only provide just protection from the retinal ganglion cells for combined treatments, just the solution of ketorolac plus the ketorolac microspheres, or the solution alone in this model, compared with the microspheres loaded with ketorolac, demonstrated the importance of the injury. So probably in the animal models, we have to take into account that the damage in glaucoma is high when the increase of IOP is observed, and then followed by a chronic degeneration. So this is, I think, this is a very important topic to have to take into account, and we can we can test different strategies, I think, for just to test novel drugs and then delivery systems for the treatment of this disease and other diseases affecting the retina. So I would like to thank all the members of the group and all the collaborators that have contributed to these works. Thank you.
Brilliant. Thank you, Rocío. That's a lovely talk. Thank you very much for that. Any questions from anyone? Oh, I'm just going to stop the share so we can all see. Okay, well, if I may start then. So all of your glaucoma treatments, they're all neuroprotective treatments, but obviously you still have developing glaucoma over the period of time. So do you think it would be wise to combine that with drugs that will reduce the pressure?
Yeah.
You see more neuroprotection if you were to give your neuroprotectant and reduce the pressure at the same time? Yeah,
I'm totally convinced. And for neuroprotection, I'm convinced that multi-loaded or multi-pharmacological approach are necessary, for sure. Mm-hmm. And also that maybe we have to differentiate between acute injury and chronic injury. And it's possible just to combine both. According to your results yesterday about caspase,
yeah,
I think we could do it.
Yeah, I agree with that actually. So I mean that's the other thing about what you're showing here. Glaucoma is a chronic degenerative condition, so it takes a while to develop, and then you give your treatment. So when we think about patients, then Richard might be able to come in here. When we think about patients, if you were to so most patients don't know they have glaucoma until a little bit later. So with the treatments that you're proposing then, do you think if people have already established glaucoma, would they still be neuroprotective?
Yeah, I think so. I think we have to push just for protecting the retinal structures just before the disease. Mm-hmm. And then we have to develop different strategies for personalized therapy in this kind of patients. It's different that when patients has an increase of IOP during long term, is different than they have just only an increase of IOP and then normal values. So we have just to work for different strategies.
Thanks, Rocío. Anne, you've got your hand up.
Hi, it's a great talk. I was just wondering about with these particles. Is there a limit to the mix of molecules, the number and also the type? I mean, do they have to be of the same type of molecule that you could encapsulate together, or can you make different mixes? And what are the limitations around that sort of multifactorial delivery?
Yeah. Thank you, Anne, for your question. Yes, we are encapsulating until now up to 4 different agents, and we have developed a technology just to encapsulate biotechnological products and low molecular weight substances in the same device. Mm-hmm. So we are working on that in order to administer the low amount of biomaterial with the best profile. So we are working on that.
Thank you.
That is possible. You're welcome. You're welcome.
Question from Rocío, a question from one of our ESRs. Do you think that the size of particles would affect the drug release rate? Do you think the size of your particles
of course
will affect the release rate?
Yeah, of course. Of course. This is the reason why we choose among the different size, just one that can be easily injected and providing just a nice profile. Yeah.
Thank you. Okay, another question in the chat. what is the best way to identify the effective concentrations of combination drugs? For example, in your study, you have different concentrations of DEX, melatonin, and coenzyme Q.
Yeah, this is a very good question. It's very difficult just to answer that question because in glaucoma, we have a lot of empirical data. We know that it's multifactorial. We know that there are different targets, but hopefully we could know in the future what is the best concentration. But in my opinion, probably with very low doses, if the therapy is combined, will be feasible.
Rocío, a final one from me, I think. So obviously we can protect retinal ganglion cells with lots of different things, but obviously in glaucoma you have axonal degeneration afterwards. So do you think that if we were to protect the ganglion cell, we'll stop the axonal degeneration, or do you think we need other treatments for the axonal degeneration that you see?
I think with a combined therapy, we could protect just the optic nerve, the photoreceptors. We have done several proof of concept just demonstrated that the photoreceptors can be also protected, and all the extractors.
Okay, well, thank you very much, Rocío. That was fantastic. Thank you so much for that very stimulating talk. Okay, we'll move on to.
Thank you.
Thanks, Rocío. Our next speaker is Dr. Rami Ghandam. He's at University of Glasgow at the moment, and we're collaborating on this particular project. So I'll hand you over to him, and then he can tell us what he's doing.
Thank you so much, Zubair. Thank you so much for the invitation, and congratulations on your recent promotion to the rank of professor.
Thank you very much.
Well deserved indeed.
Thank you.
So my talk is actually going to be quite different from the rest of